Great pleasure to be here today to talk to all of you. It's really good to be able to tell you about the research we've done, and I'm sort of pretty thrilled to tell you about how that's been accelerated with some early access to the Xevo MRT. So probably some of you know that my group has been involved in trying to find metabolic markers for Parkinson's disease using an unusual biofluid that is sebum for some time. And those of you who know anything about diagnostics know that it's quite a hard journey, but recently we've had some good, promising results, so I'm getting a chance to talk to you about it. So let's first of all think why it is that we might need to diagnose Parkinson's disease. It's a progressive neurodegenerative disease, and it's the second most common age-related disorder after dementia. It's the fastest growing neurodegenerative disease globally. A lot of that is because we're getting older, and some of that is because there's improved diagnosis. But some of it is to do with the use of pesticides. In the main, Parkinson's disease is diagnosed after the motor symptoms that we all think of about it appear. And if we look at this image, If we look at this image here, we can see that by the time things are diagnosed, by the time we've progressed to clinical, the substantia nigra and dopamine receptors therein have been really damaged, sometimes to up to 70%. Whilst we think of it as a disease that affects old people, 4% of people that are diagnosed globally are under the age of 50. And for that age group in particular, and actually particularly for women, diagnosis can take up to 8 years. And what that means is those people are being unnecessarily treated. If you're stiff arm, one of the early symptoms, you stop swinging your arms when you walk, one arm becomes stiff. So you're then sent to have frozen soldier surgery. You may have gut problems, you may have sleep problems, you're put on sleeping pills, you're given gut treatment, and all of that is Parkinson's. That prodromal stage with those early symptoms can last up to, well, certainly up to 15 years. And our group and many others want to understand that progression and want to find markers for that progression to help with what we call timely diagnosis. At the moment, there are no drugs that could treat people pre-motor, but in development, there are neuroprotective agents. There's currently about 84 in development. And so being able to diagnose in a timely fashion will perhaps accelerate the uptake of those and our understanding of those. So there isn't a test available yet for Parkinson's disease, and this figure here shows the symptoms. I think the symptoms that are on the left-hand side are those that most of us associate with the disease, so that's the motor symptoms. But those on the right-hand side are things that, unless you know someone with Parkinson's, you'll probably be less aware of. I've already mentioned gut problems, sleeping problems. Pain is a really significant part of this disease. And then, interestingly, as you'll see, a loss of sense of smell. The diagnosis, therefore, is based on the observation of motor symptoms and taking a medical history. And there are different stages that have been developed such that we can start to have markers for progression as well, but this is all based on clinical observation. If you're an expert movement disorder specialist, your diagnostic capability is about 80 to 85%. So that means there is some underdiagnosis. The FDA improved the DAT scan in 2011, and that is useful, and there's been some wonderful new work in imaging contrast agents, but that's only really used in difficult diagnostic cases. It's expensive, and it's not always accessible to the individuals involved. There's been some very interesting recent progress with the use of seeding assays using pre-seeded alpha-synuclein, but that mostly relies on using cerebral spinal fluid, and that is not necessarily a biofluid that really we should be taking to try and screen for a disease. There has not been as much progress as certainly the Parkinson's community would like on defining observable, testable progression markers and on early diagnosis. So I didn't want to solve all those problems. This lady came into my life. And she's called Joy Milne, and she had a very interesting story. Her husband was diagnosed with Parkinson's disease when he was young, so he was in that early, young onset Parkinson's disease cohort. But about 12 years before he was diagnosed, Joy noticed that he smelt differently. She told him to wash more. He was not very happy about this concept. And life carried on, and of course other symptoms came, and he was eventually diagnosed. Some years after that, she realized that all people with Parkinson's had the same odor. And she came to a colleague of mine who was giving a talk on his work on Parkinson's, and she challenged him to do something about this. She said, there is an early marker, it's the odor. What can you do about it? Well, he's a clinical biochemist. He really didn't know what to do at all. He looks at stem cells. But he rang me up and he said, Purdy, could you or could mass spectrometry diagnose or find a volatile? And I said, well, yeah, sure, but really? Do you really think she can smell disease? So we had to think about this a bit, and what we did was decide to have a method where she couldn't see the observable symptoms of Parkinson's, the motor symptoms, but she would be able to smell the odor. So we basically gave Joy some T-shirts to smell that had been worn by people with Parkinson's disease. We had 6 people who had Parkinson's disease, and we had 6 people who were control subjects. We cut them in half, so Joy actually had 24 T-shirts to smell. And she was entirely correct about the 6 individuals who had Parkinson's. Not only that, she was able to put the people back together. T-shirt number 12, that's matched with T-shirt number 16, that's the same person. It's remarkable. One of the T-shirts, which was in our control group, she said smelt of Parkinson's, and she grades from 1 to 4, with 4 being a very, very strong odor, and she said it was near 3. It was really quite strong. She was very definite about it. So false positive. Except it wasn't. That individual came back and told us 9 months later that he had just been diagnosed with Parkinson's disease. And so again, as she had done with her husband, she was able to pre-diagnose the disease before it had been clinically diagnosed. So that was remarkable. I cannot tell you how extraordinary it was to have this. You know, you just think, wow, someone can actually smell a disease and find it even when the person hadn't been clinically observed. Actually, the most remarkable thing about us using T-shirts was that we found out that the odor wasn't strongest in the armpits, which is what you'd think, right? You'd think it would be like that. It wasn't. It was in the area underneath the neck. And that's an area where we all collect a lot of sebum. We have it coming from our hair, and also, I don't know about any of you, but it's pretty difficult to wash, you know, that part of you, so you can sort of scrub. But anyway, it's rich in sebum, and that then led us to start to investigate this biofluid and to see what biomarkers we could find from it. So we set about to collect it and see if mass spectrometry could do what Joy could do and distinguish Parkinson's from control subjects. We've sought to develop a method to sample from people, and there's some images here from the sampling packs we send out to people at home, and then there's a picture there of the sort of pack that we send people. So we do everything in regular mail. There's no need for cold transport or cold storage, which is quite remarkable. We send Q-tips, wooden Q-tips, and we send gauzes, we send some gloves, and we send some jiffy bags for people to send the samples back to us. And we advise people to either self-sample their long-handled Q-tips or to get someone to sample from the mid-back. For thermodesorption gas chromatography mass spectrometry, we use gauze because it's a larger surface area, but we started doing that for LCMS, and we were using 9 mils of solvent per extraction, so we moved to the Q-tips, and they're actually much better, easier to use. We also sought collaborators, and I'm happy to say we have collaborators all over the world. We're involved in a very large study which is run out of the H.T. Chan Center in Harvard University with Cortese and Alberto Schiro, and that's sampling from nurses in the US who have prodromal indicators of Parkinson's. So these are nurses who are just regularly, sending samples into hospital labs all over the country, and these are the cohort who have suspect Parkinson's, so we're looking for them. We have samples from all over Europe, and we're also involved in a very large study that's sampling people from 10 sub-Saharan African countries. Our current findings are this: with thermodesorption GCMS, which is most similar to smell, right? We find about 600 volatiles on everyone's sebum, and about 5% can predict PD now with 97% sensitivity. There is a prodromal, very likely prodromal form of Parkinson's called REM sleep behavior disorder, and that has a different volatile signature, and we found that. With LCMS, using different platforms, we find about 25,000 features, and about 10% of those are significant for differentiation between Parkinson's and control. We never thought, but we were able to do some really detailed pathway analysis to learn about the pathways that are disrupted during the disease, and there's substantial lipid dysregulation and alteration of the carnitine shuttle. We've also used paper spray coupled with our mobility mass spectrometry, and we can really now see some very, very long lipids that are substantially upregulated in people with Parkinson's disease, and that's a very fast assay. One of the cohorts that we've been working with a lot is based at Oxford University. It's been there for more than nearly 15 years now. 1,000 people were recruited, and they've been followed up at regular intervals. There's very good matched controls, and there's also a large cohort of people with REM sleep behavior disorder, which is a condition where if you had 100 people, within 10 years, 75 to 80 of them will have converted to Parkinson's. The conversion rate is about 8% a year. And there's also relatives to study. In the work I'm going to show you, we've taken a subset from this well-studied cohort, 76 samples with Parkinson's, 78 with RBD, and some matched controls. We mostly, for this, we sampled these people at home. We sent the sampling packs to Oxford, and they sent them on to them, and we're blinded to it. And this is led by Michelle Hu at Oxford. We increasingly look to my predecessor at the Michael Barber Centre at Manchester, who some of you may know, Simon Gaskell, to think about the way we approach analysis. I think one of the great things about omics and mass spectrometry and everything is how many features we find. But actually, you talk to a clinical mass spectrometrist and they want one qualifier and one quantifier. So what is the point of having all those lists of metabolites and all those lists of proteins? Actually, we want robust features that can be measured again and again. And so what we've done with the Xevo MRT, it was very kind of coincidental because this was the way we were thinking too, is to think about shortening the runtime and seeing how much information we can get. We've also run a subset of those samples on a IMS cyclic because we've seen separation with our mobility for some of the isometric and isobaric lipids, so it helps us to do that. And then we've looked at the features generated using a conventional progenesis-based pipeline and also a new faster, robust pipeline. We've then built a predictive model and translated the markers for targeted MRM. So Simon had a very useful way of thinking about this sort of approach, which is shown here, just enough diagnostic information. Right? This is really what we want, and it's what we want out of the way we do things generally and what we've done with the MRT. So here's lists of the different chromatographic conditions we've taken. So our conventional metabolomics on the cyclic is a 22-minute gradient using a Acuity I class. And then on the Xevo MRT, we've taken what we call routine. That's what Lee Gethings, who was really instrumental in doing this, he said routine. I was like, wow, if routine is 12 minutes, we're in good shape. And then high throughput, which is 7 minutes. And the really important thing about these shorter run times is that that's the sort of runtime you'd put onto your triple quad assay, right? So any chromatographic conditions that you develop, you can translate more quickly. Same solvents in all cases. Here's just showing a bit more detail about how we do those gradient profiles. So there's a bit to just try and push the lipids out. And we've already learnt something about this instrument, but just to tell you, all the data I show you is positive mode electrospray. We've got the opportunity to have two different scan rates, one at 10 Hertz, which is what we use in the routine, and in the high throughput, mode, we're running at 20 hertz, which is pretty amazing when you consider that 4-meter ion path they're going through. Collision energy ramp and source and sample conditions as shown there. Something that hasn't been mentioned is it's really quite wee, this instrument. It's really not very big, and you know, that's a nice thing, and you see it there in profile. So here's some comparator chromatograms. This is our conventional cyclic 22-minute gradient. This is the routine on the Xevo MRT, and then this is pushed to the 7-minute. And these are all rich with diagnostic features. And why are we doing this? Because you want to get things done quickly, particularly if you want to take this to screening or to some sort of targeted approach. The mass spectrometry data from this instrument is pretty remarkable. So this is a triglyceride lipid from slightly later, and this is in the routine chromatographic run. It's about 12 minutes, and that's coming through at about 8.5 minutes, and that's a representative lipid that we see. And even when we go to high throughput, now at twice the frequency, so if you can see the intensity, it's a teeny bit lower, but not really. I mean, sorry, it's higher because we're sampling twice as fast, but it's really commensurate, and the mass resolution isn't compromised at all when we do that, which is pretty remarkable from a small instrument that's working pretty fast. So we can compare the peak capacity that we get with our cyclic, around about 55, and the resolution we get there on these sorts of samples is about 45,000, with that we're getting with the MRT. And you can see there's a significant improvement in all cases there, even on that 7-minute gradient, so about a third of the speed. So we've taken two ways of processing, like, you know, let's take some really interesting samples, put them on a brand new instrument, and let's just use a different processing pipeline too. But that's reaped some pretty good results. So we've used a routine progenesis approach where we take the data, deconvolute, de-isotope, do alignment with QCs, peak picking, and then identification with statistics. We've also taken a novel method, working very much with Hans Vissers at Waters, which takes different modules out of the progenesis pipeline, but is very much faster, and to an extent is signal agnostic, not signal agnostic, but data agnostic, and allows us to really pull things through much more quickly. In the end, also using R to generate models for prediction. So here's just an example of some of the data we get from cyclic. This is LCMS data deconvoluted with progenesis QI. Do all of our peak picking and alignment and area normalization, and this takes quite some time, and all of our samples are referenced to pool QCs in both cases. You can see these are rich data sets, and that's not even looking at the ion mobility data with, as I've said already, around about 2,400 features. We then take features that are absent in more than 10% of the QCs out and take those where we've got big variation in QCs also out. This leads us to around about 12,500 features, which we then are able to robustly detect throughout. Some of those features are a particular interesting thing of doing sampling where we sample. Anyone want to guess what those are up there? Because they're sort of things you've probably seen before. You get them more with women, it's shampoo. So we see that coming through, but that's okay, that works as quite nice markers. So we also have applied progenesis methods to the new data, but I'm not going to show you that at the moment. But the modular pipeline is producing similar sorts of sample numbers, so from total features and then down to scaled features. The difference in the two numbers there is this was us looking from 50 to 2,000, which is what we tend to do for our cyclic data, and this is looking just up to 1,600, which is more commensurate with what we did on the MRT. And you can see there that even with those shorter run times, we're still getting an enormous number of features. And just to remind you all, we've taken a Q-tip, it's been rubbed on someone's back, and we're analysing it, and we really do have a lot to work with. We then take those features and start to cluster them, and you can see there's some pretty good clustering going on, and here's an example of some of this is just doing leave one out to look at the robustness of features. And in this was based on cyclic data actually, we built a model based on 3 features, which gave us and that was only a subset, it was only 15% of those 200 samples we run there, which gave us fairly good confidence. We then reran that on the MRT data, and we have very good predictability. So on a test set, we now, based on this sample size, we have 88% sensitivity with what we've achieved. So we were pretty pleased about that. It's great when you do things on 2 discovery platforms because you can go and check that you've got the same feature, and indeed we do. So the data here is showing data from the cyclic data from the cyclic is at the bottom and data from the MRT. You can see there's a little bit of crossover there, but of course we can pull out those features. And those were enough for our predictive model. So top 7 was enough for that predictive model I just showed you. And this is then transportable for Skyline for targeted assay development. So what's the prospect of using this for a test of clinical utility? That's why we're doing what we're doing. Well, I've shown you that sebum is a very rich source of diagnostic biomarkers. The sampling is just non-invasive. It's really incredibly easy. And actually, we all got used to that sort of thing with COVID, but it's a lot better doing that than shoving something up your nose. It can be done at home, and samples can be transported and stored under ambient conditions, which is really helpful. All of this, and anyone working in translation of clinical omics to anything that's going to be useful, needs to work along with clinicians, and we do that a lot. Our ability to reduce any false positives or false negatives increases dramatically if we have clinical markers, and we do. So what I've shown you is our work curves for just the mass spectrometry data. If we add in one of the markers that we know clinically of those people, then we have pretty much 100% accuracy. Our aim is to work to stratify this disease and how it progresses, and to use biomarkers to inform clinicians and improve the outcome for patients, particularly at the early stages, but also as the disease progresses. We think that there's a really good opportunity, as I've told you before, to use this method along with trials for neuroprotective agents and other therapies for at-risk groups, so people who have familial or genetic risk of getting Parkinson's disease. Being able to translate to an MRM method for clinical biochemistry labs, because that's what they use triple quads, is really facilitated by taking this sort of approach. And in our hands, it's been really facilitated by access to the Xevo MRT. The great thing, as I've mentioned, is that we can map that discovery gradient right onto the targeted gradient. And this new pipeline has allowed us to rapidly assess putative features. I think an approach where you take 2 discovery platforms, you run the same samples on them, and then look for features is got to be the way forward. And we found it pretty exciting to be able to do that. Just to acknowledge, there's Joy and Les, who's no longer with us, but that was them in happier days, and a lot of the clinicians that we've worked with, and you for your time. Thank you very much.